Thanks all of you to come over this afternoon. Very humid weather. My link with David is that I worked with him for within an exchange program for 4-5 months, something like that, doing the analysis or helping out with the analysis, RNA sequencing and mass spectrometry. But what I'm going to talk about today is some other work, the integration of one specific kind of sequencing technology, which is called ribosome profiling, and the different biological questions or applications we perform with this technology. There's 2 different chapters, 3 actually, in my talk. The first one is more general. How can ribosome profiling help out integrating with mass spectrometry to identify novel proteoforms? The second topic will be on putatively coding small open reading frames and how we can identify these. And then the third one will be on Epitranscriptomics. I'll come to that later, but we also want to integrate this with ribosome profiling. So let's start off with the quest for novel proteoforms. First thing I have to explain here is what is a proteoform. I guess some of you already know this. It's like the grouped variability at the protein level. There's a lot of different ways how variability can be introduced at the protein level. You have genetic variation. You have, to a lesser extent, also RNA editing. There's a lot of alternative splicing going on at the transcript level. There's also a lot of alternative translation initiation going on. This has been described in depth with the advent of this ribosome profiling technology, mainly that you have translation initiation in the 5 UTR regions that can yield small open reading frames in the upstream region. You can also have N-terminal extensions. These are the blue bars, whereas the gray one is like the canonical translation product. And you can also have N-terminal truncations. And something else that was found is that translation does not always start at cognate start sites, so at methionines. To a lesser extent, you can also have stop codon read-through and frame shifts. And then another thing that adds up to the variability at the protein level is the modifications. There's a lot of modifications that make that certain proteins become more stable, have a certain function whatsoever. So this bundled thingy here is called the proteoform, and the different levels where variability can be introduced. Okay, everybody knows Wally. Sometimes it's hard to find Wally, and that's exactly what mass spectrometrists want to do. They want to match their fragmentation spectrum to a peptide in a database. This database can become very big, so it's sometimes very hard to match. By now, you all should have found Wally. I gave you at least 10 seconds, so it's just to point out that it's sometimes hard to find Wally. Wally is here in the picture. And this is already true if you look for like the regular Wallies, the canonical proteins, the stuff that we all know. But what if we look for extensions? They have a walking cane. Or what if we look for truncations? Or what if we look for Wallies with one blue stripe instead of all red ones? Or what if we look for very small Wallies? If this information is not in your database to search against in mass spectrometry, for mass spectrometry data, you will not be able to identify these Wallies. Peptides, proteins, of course. There has been a lot of integration, also in David's lab, RNA sequencing and mass spectrometry, building custom databases from RNA sequencing experiment, matching same samples. A lot of this has been done in cancer research. to identify novel alternative splice isoform, to identify variant peptides. As I said, a lot in cancer research has been going on there as well, also looking for fusion peptides. Specific biomarkers for certain cancers can be done with that. Most of the time it boils down to the fact that you create a custom database built from this information you gather from sequencing. So you increase your Wally space in a way. There has been also a lot of recent large-scale proteogenomics with all their concerns. I mean, we have the human proteome studies. Akhilesh Pandey's group and Kirster's group provided us with very thorough human proteome maps. And also, ENCODE had a proteogenomics part in it, CPTAC as well, and also the chromosome-centered human proteome initiative had a lot of proteogenomics. But we tend not to work with RNA sequencing to create our custom databases. We want to use ribosome profiling. So what is ribosome profiling? You don't sequence the whole mRNA. What you sequence is really the stretch which is captured within your ribosome complex while it's translating. So in a way, you just measure or you get a snapshot of what's being translated at that moment in your sample, cell line, tissue. So basically, you halt your ribosomes while they are translating. You perform a nuclease treatment so that the overhanging mRNA stretches are removed. You know that the captured fragment is still protected. You recover these fragments, and then afterwards you convert it to a sequencing library, and then you sequence. You have two different flavors of this ribosome profiling. The first one is just halting everywhere so that you have a complete snapshot. This is actually the blue signal. This is a transcript. So the regular or the baseline experiment would give you a signal as this. So it gives you a signal all over your coding sequence. And then you have an extra one where you have an extra antibiotic treatment, and that makes that these ribosomes are really accumulated at start size. So basically this would give you a nice crisp peak at translation start size. So using these two types of information, you are able to delineate the open reading frames and hence also do the translation and hence also compile a database that you can use for mass spectrometry identification. I work in a bioinformatics lab, so of course we build a tool to do this process. There's several steps. So of course you have the sequencing of these ribosome protected fragments using available sequencing technologies. An important step is also the quality control. Before we move to creating custom database, we want to make sure that the quality of our data is well enough so that we can actually build this snapshot of what's being translated. We map all the reads, the ribosome protected fragments, using available mappers. Most commonly used are, or splice site aware ones, are star and top hat too. We are also able to extract variation information from this sequencing pool that we can also introduce in our custom database. And very importantly, and that was the red peak, we are able to pinpoint where translation starts. We pour all this information into a relational database, and then we move towards the amino acid level, and then we do the translation, taking into account all these awarenesses that we gathered from this ribosome profiling information. And afterwards is mass spectrometry identification. We created this tool in Galaxy. For those who are not aware what Galaxy is very popular in sequencing research groups. It's a web-based tool which allows you to do high throughput analysis. There's three different sections here in this web interface. The left pane gives you all the tools which are available. As I said, there's a lot of genomics, transcriptomic, and also proteomics tools already available, which are already implemented in this environment. The middle pane gives you what you are doing at this point. And at this point, I'm just depicting a workflow built out of tools from this left-hand section. And here is more detailed input variables for these specific tools. Or also, if you're really running something, this would give you a history of output files, the result files. So as I said, the first thing is mapping. And there we already spit some information that you can use to visually inspect in a genome browser way your ribosome profiling data. I'll show some plots later on. Quality control, so metagenic functional annotations. Where do these RPF fragments map to? Most should map to exonic regions, of course, because that's where you tend to have translation. The two upper small boxes is basically pinpointing the transcripts which have RPF coverage. So the transcripts that should be retained. And then the upper one, the clear red signal, is the TIS calling, so the translation initiation site calling. Based on this information, already pointed this out, you can build your translation database, whether or not you also include variation information in there. And then afterwards, you can use existing tools, and we mainly use Search GUI and Peptide Shaker, because these come from a lab which is very close to our lab, the Compomics group of Leonard Martens. And Search GUI is just a graphical interface towards different search engines to launch them together. And Peptide Shaker takes all this information and tries to bundle everything and does protein inference and interpretation of your data. So we did some validation study to see whether adding up custom information from ribosome profiling to your search space gives you extra identification. So we did two types of different mass spectrometry experiments. The first one is a regular shotgun experiment. The second one is the N-terminal cofaradic. This mainly only tries to identify the N-terminal peptides, which will be very useful because we also pinpoint the translation start sites with ribosome profiling, so we can overlap this information. We did that on two different cell lines, two different species, a human colorectal cancer cell line and a mouse embryonic stem cell line. This is just a plot, although correlation plots don't always say everything, to see whether quantitatively ribosome profiling does correlate with spectral count-based measurements of mass spectrometry. So the x-axis gives you these spectral count-based measures, and the y-axis gives you these RPF count, so ribosome profiling-based information. And as you can see, both for human and mouse, you have fairly good correlation coefficients, better than the ones you would obtain with matching RNA sequencing information. Okay, so we use this extra information to search against from ribosome profiling, and what do we get? If we look at the shotgun proteomics experiments, in gray is what you would identify using only a SwissProt database. This is at the protein level. In green is what you would identify actually by using this custom search space. So as you can see, the overall identification rate improves, and there's both new identifications, which is the light green pie part, and then the dark green pie part is those identifications which have better proof or better score because they have an extra peptide, tryptic peptide identification in their protein. If we look at the N-terminal cofaradic data, Again, in gray is what is known, the annotated translation start sites. And then you can clearly see that next to the canonical ones, you also are able to identify different proteoforms. So for example, in red you have truncated proteoforms. In yellow you have extended proteoforms. And something else you can see is that these small pies represent the both near cognate start site translations and cognate start sites. So you see for the extended proteoforms, you have a lot of these that start at near cognate start sites, which is quite remarkable. And what we could also validate from ribosome profiling studies, previous ribosome profiling studies, is that sometimes for certain transcripts you have multiple translation start sites. But this is not visible on this picture, but I'll come to that later. So just a few examples. To the left you have multiple sequence alignment of the custom form, the upper one, and the lower one is the Swiss plot. And then to the right you have the annotated spectrum. So the first one is a peptide identification within the 5' extension of the protein. The second one is a peptide which has a single amino acid variation. And then the third one is an example of pinpointing the exact translation isoform based on this information. And this is an example, it's the HDGF gene. We could also identify this for human, is an example of, this is the UCSC browser. What you actually can see is the upper part, so the yellow is the signal of the baseline ribosome profiling experiment. So you would see a signal all over your coding sequence. The blue line is the special treatment. So you would see a clear peak at start sites. Here you have a magnification, and you can clearly see that at these positions, this is the canonical start site of the protein, but you have 3 positions where you could have an extended proteoform. And for 2 of these, we were able to identify the N-terminal peptide as well. So, to conclude, first part, adding up custom information from ribosome profiling in mass spectrometry identification really helps out. Second part would be on coding of small open reading frames. The last few years there has been an increased interest in small open reading frames, mainly because there were some small open reading frames that have been functionally characterized. So whereas the canonical bioactive peptides are cleaved from larger precursors, mainly they have a single peptide as well, so they're going through the secretory pathway. These micropeptides are translated directly from the small open reading frame, and they lack this signal peptide, so they are released in the cytoplasm. That's what people believe. And as I said, the last few years, already more than 10 years ago, there was some evidence in plants as well and insects as well. But then the tarsal less was the first peptide that was small open reading frame peptide that was identified in Drosophila. And then only 1 or 2 years ago, there was also some identifications in human and mouse having impact on calcium pump, muscle contraction. Again, we're a bioinformatics group. So the first thing we try to do is, since we have the complete genome, we try to find locations in the genome where coding small open reading frames could reside. We use 2 different model organisms, so fruit fly and the mouse. We used the fruit fly to build a strategy, and then we extrapolated to mouse, mainly because for fruit fly we had smaller genome, of course, and we also had only a few examples to validate our findings. Again, this is more bioinformatics stuff. How did we came to this? First of all, one can imagine there's a lot of small open reading frames in the genome, so one has to filter at some point. So our first filter would be using an existing tool. I know it's not perfect, but that's what we used, which is called SORFinder. It uses a hidden Markov model to really pinpoint coding small open reading frames. So high coding potential small open reading frames. Afterwards, we used multiple sequence alignments to really come up with a measure of how well are these sequences conserved. And then afterwards, we used support vector machine techniques to somehow rank these small open reading frames on coding. Visualization was done in an in-house browser, and this is, for example, visualization. This is the conservation. The higher the bars, the better conserved. This is based on the fastcons measure. This is just to pinpoint at the antisense a small open reading frame, and this gives you the ribosome profiling information, the baseline ribosome profiling information. We used ribosome profiling to validate these findings. So basically we had an in silico way of doing things, but then we wanted something to match against. At that point we did not have any mass spectrometry ready to do so, but so we used the ribosome profiling to validate these in silico findings. So for mouse, we came up with a list. For Drosophila we came up with a list, and well, the first one, of course, was the tarsal-less. Within the top 10 of our list, there were some good candidates, actually, because the first one was the one that was described by Kageyama's group in 2010. And then the second one, after we did our prediction pipeline, I think the year after, it was described by Magny et al. That was also the one that has an impact on muscle contractions. Just to say that apparently this in silico method works quite well. So we also extrapolated this to mouse. Of course, the genome is way bigger. So you have the different regions where these small open reading frames can reside. And then afterwards, we overlapped this with ribosome profiling. We came to a list of candidates which could be further investigated. This is just a plot, the chart again, to show you these small open reading frames. They can reside in all kinds of functional regions. Since the biggest chunk of genome is intergenic, the more small open reading frames will reside there. Same with intronic, exonic, and non-coding. But if we look further down this line, so if we include these coding predictions based on conservation and the support vector machine, you will see this shift. So there's a huge or a much bigger part than previously in exonics, just to point out that actually this prediction pipeline also works for mouse. But here, validation is still underway. And at that point, we decided to use a different method and start the way around. So we wanted to start from ribosome profiling rather than in silico, and then from the findings from ribosome profiling, we wanted to do this the conservation measures again, and also extra metrics. For example, this floss score was described by Ingolia, just to give you an idea, only based on ribosome profiling information, if this specific open reading frame is coding or not. And this measure is actually based on, you have these stretches which are captured within the ribosomes. On average, these are 30 base pairs long, but there's a distribution. So this is just matching a distribution of known coding regions and the length of these stretches with what you want to investigate. So the better these two distributions overlap, the higher the proof that it's actually coding. So basically, these steps are the same as we used in the proteoformer tool, the quest for novel proteoforms, and these are the extra ones. So introducing phylogenetic conservation using the phylo-CSF tool, and also introducing extra metrics based on ribosome profiling to see whether this is actually coding or not. Again, we port this in this Galaxy framework so that wet lab researchers can also apply these tools. And this is what comes out. What you can see clearly is, again, that these small open reading frames can reside in all regions, 5' UTRs, exonics, intronics, 3 UTR, non-coding regions, and intergenic. But then there's two things you can do. Either you just base your identifications only on ribosome profiling, or you go for matching mass spectrometry, which we also did. If you only use ribosome profiling, you can filter on the coverage. The better covered with ribosome profiling, the more proof it's actually coding or not. You can prove it on translation, and therefore we use this floss score, for example. Or you can use conservation metrics to really say if it's coding or not. Again, we wanted also to have mass spectrometry proof, but most of the time, these small open reading frames are low abundant, and you somehow need, and also translated or expressed at several times, specific time points, and also specific cells. So you really have to enrich and use extraction and extra separation protocols. So we tried different kinds of stuff. We did exosome enrichment, we did synaptosome separation, or we also analyzed specific cell lines where we thought some of our candidates would be expressed. There's also a lot of extraction, enriched extraction going on. There's also a lot of small peptide protocols, extraction protocols, for example, the heat inactivation, for example. And you can also use very long separation columns to really have as much as possible from your sample. So at all these levels, you can optimize. Another thing you can do, rather than going for specific analyses or mass spectrometry stuff, is really bulk search everything which is available. You have this PRIDE database. PRIDE is a database, a public repository for mass spectrometry information. What we also did is we built this custom database of these small open reading frames, and we use this to match against all the mass spectrometry data which is available in PRIDE. This is still ongoing. A few examples of what we already found so far. Within a long non-coding, we have a small open reading frame, quite well conserved. It also has a very good ribosome profiling coverage. As you can see here, again, it's the red and the blue signal. Blue signal is signal all over your coding sequence from ribosome profiling, and red is start site, so it also has a very good ribosome profiling coverage. This particular one does not have, so far, mass spectrometry evidence. Then another one, also in a long non-coding region, we found this small bugger in PRIDE reshake analyses. So in more than one data set in PRIDE, this sequence came out of that as a valid identification. And then another one is an example of MKKS gene, where within, as you can see, it's antisense, so it's this direction. This is the canonical start site. Within this upstream region, you have an upstream open reading frame, which is well conserved between mouse and human. And it also has nice ribosome profiling coverage, and it also has nice mass spectrometry validation as well. This came from the synaptosome enriched data set. But one has to be very careful in trying to identify or at least interpret these small open reading frames or the identifications. Sometimes an upstream open reading frame can point to a 5' extension of your protein because of misannotation, because of mutation point. This can happen. So you really have to take care of that. sometimes we tend to go for a two-way identification search. So basically first search against what's known, Swissprot, and then afterwards we search against the new stuff that we come up with, small open reading frames. Sometimes an identification against the new stuff actually points to something which just fell below threshold identification-wise, but is very abundant in your reference database. So you must really be careful. Sometimes you have stuff that's not in Swissprot, but can be re-annotated. It is in translated TMBL, but based on these searches you can re-annotate. Or sometimes this identification can point to a variation from a reference peptide, which is quite, that can be the case quite often. So for these few things, you can actually search multiple databases of known sequences to really make sure that the one you find is really new. So this is something that you have to include. You also have to include CrabDB is like stuff which, contaminants which occur quite often in mass spectrometry. So you have to include this as well, just to make sure that what you identify at the last level is really new. Again, it can be variants. There's isobaric masses, near isobaric masks. Two amino acids can point to, are sometimes almost the same as one. There's a lot of modifications which can make that you actually identify modified reference peptide rather than a new one. This all holds for these specific small open reading frame studies, but also for proteogenomic studies in general. And these guidelines have been well described in a review by Alexei Nesvitsky. We're, again, trying to automate all these validation steps into our pipelines as well. All this information on the small open reading frame stuff, all the metrics, phylogenetic conservation, rescanning against PRIDE, public repository of mass spectrometry data, also phenotypic mutation points co-localized with small open reading frames. All this is bundled within a website which is called source.org. The third part of my talk will be on, and last part will be on Epitranscriptomics and how we try to merge this again with ribosome profiling, because that's really the red line within my talk, ribosome profiling. Everybody knows this, more or less. DNA is transcribed to RNA, translated to protein. I would like to focus on the top part here, the epigenome. I mean, next to the sequence itself, you also have modifications. The epigenome is already established. It's known that methylation on CpG islands in promoter regions have a silencing effect on the gene itself, and it has been studied quite often in cancer research. Epiproteome, one could see the epiproteome as being the modifications on top of the proteins or the peptides, which have an impact on function, stability, and so on. But what I'm going to talk about here is epitranscriptome. It has been around quite long, actually. It's known that mRNA is heavily modified. The M6A, the N6-methyladenosine, is very abundant in mRNA. But the last few years, several research groups also were able to identify the enzymes that make that these methyl groups are added or removed from the mRNA, the adenosines. So this points towards there must be something functionally going on here. And also, with the advent of new sequencing technology, one is now able to try and establish or gather a genome-wide picture of regions that are methylated at the mRNA level. It's just, you start from an RNA sample, you fragment, then you use specific antibody to enrich immunoprecipitation for these regions which hold these modifications, and then you sequence, and then you compare a baseline mRNA with an enriched methylated mRNA pool. So as I said, you do mRNA sequencing and immunoprecipitation mRNA sequencing. So basically you have overall overview of transcription and then an enriched signal of those locations which have these modifications. So based on that, you can use peak calling algorithms to pinpoint where these sites are. And then based on these peak positions and the sequence surrounding these peak positions, you can actually go and look for motifs, de novo motif finder. And you can also pinpoint the peak to the motif location. We did this on this colorectal cancer cell line again, just to have a complete picture of this cell line, because we already had ribosome profiling, we already had mass spectrometry. We also wanted to do this. We also have DNA methylation stuff on that, so we now also wanted to do RNA methylation on that as well. So this motif is known for adenosine methylation at RNA level. So this is good. That proves that it was a successful experiment. And also if we do a functional analysis, it also proves that mainly, it's already described, mainly at translation start sites and at the stop position, so in the 3 UTR, there's an enriched level of methylation. Something else that came out of our study, and this is really new, has been described, I think this year, is this specific thingy here. Methylation or demethylation at the mRNA level makes that there is a weak or an enhanced binding of a protein which is called the heterogeneous nucleoprotein C. And this has an impact on RNA stability. It has an impact on possibly also alternative splicing. So in a way, we were able to pinpoint these motifs pointing to RNA methylation, and also the other motif, which is normally co-localized in order to allow these better binding. And as you can see from here, it tends to be also within the regions that we expected them to be. And this is, but we're in the middle of doing so, this is what we actually try to do. Again, here you have ribosome profiling. And sometimes in ribosome profiling, the baseline level, all of a sudden you have these crisp peaks pointing to translation stops there. Something is going on. Something makes that translation halts there. There's a pausing site. And we want to see whether modification at the RNA level somehow have an impact on that. This is just an example. These are methylation peaks, but we want to do it, of course, at the genome-wide level to see whether there's an enriched occurrence of either both pausing sites and methylation in the same regions. Something else we can also validate is this M6A switch that I described on the previous slide. But this is work in progress. This is just mainly to see whether we have two technologies, both at the sequencing level. The one measures modifications at the RNA. The one measures something is being synthesized into protein, and do these have some kind of a connection? To conclude, these multi-omics approaches really help out. I mean, it has been proven already. RNA sequencing together with mass spectrometry. It's now also proven with ribosome profiling. Again, we're a bioinformatics lab, so we built pipelines in order to enable wet lab users to use the tools on their data. So we came up with a pipeline to convert ribosome profiling information into something which is queryable in mass spectrometry. And we also provide the same pipeline for the small upper reading frame specifically. And we also gather all this information into a web interface. And something which is still premature, but maybe these RNA methylation points have an impact on translation efficiency or inclusion of certain exon and so on and so on. But still stuff that we have to look into in more depth. Rests me to thank quite a few people. People from my own lab, mainly Jeroen Krapé, Elvis Nda, and Volodymyr Lechuk, who's working on the small upper reading frame stuff. People from the medical protein research department, mainly Petra van Damme, with whom I work together with for all the mass spectrometry experiments. She's in Chris Gevaert's group, also in Ghent. A lot of peptidomics stuff, mass spectrometry-wise, is performed by Geert Baggerman. He's in the Center for Proteomics in Antwerp, also in Belgium. This is wrong. This must have been copied. Of course, you know David Venue. He's here located in NYU Medical Center, Chibi. A lot of people from the Compomics group as well for their tools and advice on using them. And the sequencing facility in Ghent University as well for performing these ribosome profiling and these M6A sequencing experiments. And I would like to thank you for your patience and attention. Thanks a lot.
Any questions?
For your second project on the small reading frames, when you validate them with mass spec?
Yes.
Kind of two questions. One is, when you look at the ribosome, the sequencing data, your database must expand enormously, probably, and then you listed all those databases you use to search the mass spectra against. How do you account for those gigantic databases and then the statistics of finding something that's actually significant?
Okay, so we don't pile up everything together. That's one. So we go for a multi-stage strategy. So we first look or scan the existing database. These are not that tremendously large. So SwissProt, Translate, TMBL, Ensemble, and so on. And then our specific custom database, since they're based on from RNA sequencing information, indeed, you can really explode your search space because you really have to go for 3 reading frame translation or sometimes 6 reading frame if you don't have sense-specific sequencing information. But here you know where translation starts. So you only have to go for 1 reading frame. So that's already that already has a huge impact on the explosion of your data. So these data sets are quite in the same range of magnitude as the normal ones. And we also tested this. I mean, they behave similarly as well. So that's one thing.
The ribosome sequencing data doesn't give you this explosion, basically.
That's true. And another thing is, of course, if you include variation in information, this can also blow up your search space because is it 1 allele, is it 2? I mean, sometimes you have to take into account both options. Or if there's multiple mutation sites in one sequence, it's exponentially exploding as well. So one has to we only include those that we are really sure of based on mutation analysis. Because otherwise, you have this exponential explosion of your search space again. So having a dramatic impact on identification, false discovery rate calculation, stuff like that. Yep. Okay, and then, yes?
So this is kind of a challenging question, but also on the short forms, you obviously found a lot of things, and depending on where you draw the line, you have a longer list of short ones. But if you applied a criterion that, let's say you had a conservation score that was highly reliable, and one other piece of information, ribosome profiling or mass spec. Would
Okay. It's two things. You have these occurrences in enterogenic regions. And you will have these the more in species which are not well annotated, of course, makes sense. For these, you really have to have a lot of evidence. Be it, in my opinion, I would go for both ribosome profiling, both conservation, and preferentially also mass spectrometry evidence, although it seems very hard to provide mass spectrometry evidence for these. But on the other hand, because you were asking for new genes, this would point to new genes because these are really enterogenic regions. But what we also find quite regularly is small open reading frames within non-coding regions. So these are already known. They are just wrongly, in my opinion, wrongly annotated as being non-coding, because nobody cared previously looking for small open reading frames smaller than 100 amino acids or even smaller.
Right,
Does that answer your?
No.
No.
So can you give me a number for
Come
both
again.
of those categories? I mean, are we talking about, do you think we're talking about tens, hundreds?
Tens. I would go for tens.
Tens
Yeah, it's really, I mean, it's a limited amount.
in both categories?
Yes. I would not go for a tremendous large amount of new identifications, no. It's really stuff that we missed, but it will not be, in my opinion, I might be wrong, of course, an explosion as, for example, the small microRNA stuff. I think it's smaller than that. But again, I'm only human. I might be wrong.
Yeah, my question was actually very, very related. So when Weissman came out with the ribosome profiling data, they were like, oh my god, there's so many upstream open reading frames, and like 50% of the human genes actually have it. And then there were a couple of other papers that said, oh, there may be artifacts because of the inhibitors. And then there's the difficulty to validate it with mass spec data. So
It's very
my
difficult.
question was really kind of very related. Do you think, so one thing is it's hard to validate it with mass spec data because of the statistics and the complexity and
Also the
being low abundance and all these technical reasons. But could it also be that
it's overestimated.
it's actually not real, they're actually not there?
Sure.
So I was just kind of curious about your gut feeling about this.
I'm for sure I can tell that some of these are really true. I kind of see it in all the data I have on some of these, but I would not go into hundreds or even thousands of these. What I strongly believe in still is small open reading frames within upstream open reading frames, because these tend to have some kind of a regulatory effect on the downstream canonical translation product. And that is something that I really strongly believe in. And then I'm not talking about small open reading frames within enterogenic or long non-coding, but this will be larger. This pool will be larger.
And then maybe less to produce a peptide, but more to distract the ribosome.
Yes, voila, that's it. So it has a regulatory function, and it's not saying that the translation product on sich has some function. So it really focused the attention of the machinery towards another location, yes.
There are no more questions. Let's thank Gerben again.